Cells are the fundamental building blocks of all living things, from the smallest bacterium to the largest whale. Mastering this topic will provide a strong foundation for the rest of your biology studies. A cell is the basic unit of life, capable of carrying out growth, reproduction, and energy release. Every living thing is made of cells. Living organisms have two main types of cells: animal cells and plant cells. Understanding the differences between them is a key exam topic. Animal cells are compact and flexible, containing all necessary organelles. However, they lack the additional structures found in plant cells. Animal cells contain several organelles: the nucleus, cytoplasm, cell membrane, mitochondria, and ribosomes. Each has a specific role in cell function. The nucleus is the control center of the cell, directing all activities and containing the genetic material, DNA. Remember for exams, it controls the cell and contains DNA. The cytoplasm is a thick, jelly-like substance where most chemical reactions take place. It surrounds all the organelles and is a highly active environment. The cell membrane is a thin, flexible, and selectively permeable barrier. It controls which substances pass into and out of the cell, maintaining correct internal conditions. Mitochondria are the energy makers, the sites of aerobic respiration, releasing energy from glucose. Cells with high energy demands have more mitochondria. Ribosomes are protein factories, synthesizing proteins using instructions from the nucleus. All cells, animal and plant, contain these essential structures. This table provides a quick summary of the animal cell organelles and their functions. Learning each function in one sentence is a helpful exam revision shortcut. Plant cells have the same basic organelles as animal cells, plus 3 additional structures. These are a cell wall, chloroplasts, and a permanent vacuole. The cell wall is a rigid outer layer made of cellulose, providing structural support outside the cell membrane. It is fully permeable, unlike the cell membrane. Chloroplasts are disc-shaped organelles that contain chlorophyll and are the sites of photosynthesis. Root cells, which receive no light, contain no chloroplasts. Chlorophyll in chloroplasts captures sunlight energy for photosynthesis, converting carbon dioxide and water into glucose and oxygen. This process is vital for all life on Earth. The vacuole is a large, permanent, fluid-filled space in plant cells. It stores cell sap, which includes sugars and salts, and helps maintain the cell's shape and rigidity through turgor pressure. This comprehensive summary table outlines all the key plant cell organelles, their functions, and whether they are also found in animal cells. Note the 3 plant-specific organelles at the bottom. Here's a direct side-by-side comparison of animal and plant cells. Use this diagram to quickly identify which organelles each cell type possesses and which ones they lack. The key differences are that animal cells lack the cell wall, chloroplasts, and a large permanent vacuole. This absence affects their structure, energy source, and flexibility. Use the mnemonic CVW, chloroplasts, vacuole, wall, to quickly remember the 3 structures found only in plant cells. This is a crucial distinction for exam questions. A crucial exam skill is correctly labeling cell diagrams. Examiners award 1 mark per correct label, so accuracy and good presentation technique are vital for success. Follow these 4 steps for labeling success. Identify the cell type first, use a ruler for clear label lines, spell organelle names correctly, and always provide the function when prompted. Avoid common mistakes, such as confusing the cell wall and the cell membrane, or saying mitochondria make energy. Remember, mitochondria only release energy via respiration. Further mistakes include claiming all plant cells have chloroplasts, only light-exposed cells do, and mixing up the nucleus with the smaller nucleolus. Ensure DNA is located in the nucleus. Take a moment to test your recall of organelle functions. Covering the right-hand column and reciting the function from memory is an effective revision strategy. The nucleus is key for genetic control, holding DNA and chromosomes which instruct protein synthesis. The nuclear envelope has pores for substances, like mRNA, to pass through. Mitochondria have a folded inner membrane, called cristae, which increases the surface area for aerobic respiration. This allows for maximum energy production in high-demand cells. Linking structure to function is a higher mark skill. Muscle cells have many mitochondria for energy, leaf cells have many chloroplasts for photosynthesis, and root cells have none, as they lack light. Cells are microscopically small, measured in micrometers and nanometers. Remember the magnification formula. Magnification equals image size divided by actual size, using consistent units. Light microscopes show basic organelles, but electron microscopes offer much higher magnification. Electron microscopes were instrumental in advancing our detailed knowledge of cell structure. Eukaryotic cells, like animal and plant cells, have a true nucleus and membrane-bound organelles. Prokaryotic cells, such as bacteria, lack a membrane-bound nucleus, and their DNA floats freely in the cytoplasm. This is a key difference to remember. Cells can be specialized to perform specific functions efficiently. For instance, well blood cells have a biconcave shape and no nucleus to maximize oxygen carrying capacity. Root hair cells have long extensions for increased surface area for absorption. Sperm cells have a long tail and many mitochondria for movement and energy to fertilize the egg. Here's a quick revision of the 5 essential organelles and their key facts. Nucleus controls the cell, mitochondria release energy, ribosomes make proteins, chloroplasts are for photosynthesis, and the cell wall provides support in plants. Finally, review these key vocabulary terms and definitions. Understanding these terms is essential for answering exam questions accurately and achieving higher marks in biology.